My entire life, I've been basically running away from biology. I hate memorization, and I'll never understand why the mitochondria gets to be the powerhouse of a cell. But in scrolling Instagram, one post has recently converted me. It was about Chip Wilson, the founder of Lululemon, and he was opening up about a disease he was diagnosed with more than 30 years ago. And I'm not going to pretend I'm saying this right. It's Chip is 67 now, and he was diagnosed with this disease around his 30s. He was swimming in a pool, and all of a sudden he couldn't move his arm, and he couldn't control his muscles. As he started developing back pain, he actually went to get it diagnosed, and he found out he had this muscular dystrophy. And he was already in pretty good shape then, but from there he only began taking care of his health more and more, looking after his fitness, his nutrition, his diet, everything. But of course, this disease, it wasn't in his control. He couldn't really do much about it, and it's just progressively gotten worse. 3 years ago, while he was at an airport in China, he accidentally tripped, he fell, and in trying to get back up, he realized he couldn't. His left foot, his muscles, he couldn't control it. And now it's so bad to the point where it's spread to his entire body, affecting his legs, his muscles, his arms, even his facial expressions. It sucks. This disease is affecting him and his muscles, but also hundreds of other people. And Chip has recently put more than $100 million towards trying to find a cure to this disease.
And the craziest part is $100 million is actually less for trying to bring a drug to market. And to make matters worse, there's even more scarring stats proving just how difficult and complicated bringing a drug to market today is.
This whole drug discovery process, it's one that is typically taking more than 10 to 15 years to find a cure and get a drug to market, and it costs on average $2 billion. And again, it's not just these rare diseases, but things like cancer and Alzheimer's, which doesn't have a perfect cure for so many people. And in reading these stories, it reminded me of AlphaFold 2, which back in 2020 solved the protein folding challenge and made headlines.
The interesting thing about AlphaFold 2 is a lot of its success has been attributed to games. Number one, DeepMind, the company that created AlphaFold 2, has been long known for training AIs to specifically play games. And number two, the founders have previously stated that it's thanks to a game known as Fold It that inspired the creation of AlphaFold 2 as well. And I love games. I've spent most of my summer building reinforcement learning agents to play games. And
long story short, I became obsessed with this whole idea of trying to solve the drug discovery problem, modeling it as a game, and figuring out how we can apply as many AI power-ups as possible to this drug discovery process in order to bring drugs to market faster and cheaper. Before we can actually begin powering up drug discovery, we need to understand the different steps or the different levels that the drug needs to pass in order to make it to the end of the game. It starts off with target identification, where we're basically looking for the specific proteins or genes that are held responsible for causing a certain disease. Then we can go ahead and do lead generation and optimization, which is hunting for specific molecules and drugs that will either inhibit or enhance the properties of this target. We can then enter preclinical testing, which is testing in vivo and in vitro, so cell-based testing of our drug candidate or within animals. And once the drug passes these, it can enter the clinical trials, which are very extensive, 3 stages costing millions. And then we finally make it to the regulatory approvals with agencies like the FDA. We can basically think of this process as selecting some players, which are our drug candidates identified during that target identification and lead generation and optimization phase, which are then kind of kicked out and eliminated through several games, clinical trials that it participates in, until a select few winners will actually make it to the end of the process and go through regulatory approvals. Now, the only issue with modeling drug discovery as a game is it's not as simple as a game that has a couple moves. Sure, it can have a lot of moves, but in drug discovery, the chemical search space is more like 20 to the 100, which is more than stars we have in our galaxy. So there's a lot of options. But the great thing about AI is it's great at these sort of tasks. And so because there's so many inefficiencies in the current system, AI has the potential to automate, streamline, and assist researchers in multiple ways to bring this drug discovery process down from 10 to 15 years and $2 billion to cutting that in half. Now, there are many ways that AI can superpower drug design. And if you're interested, you can check out my latest article where I actually break down some of the ideas or power-ups that excite me most. Of course, we can use AI to assist with report generation, finding the best patients for clinical trials, actually coming up with drug candidates with either de novo drug discovery, coming up with new drugs from complete scratch, or constraining it to a specific chemical search space by guiding it in a specific direction. There's a lot happening in this space. People have applied CNNs, autoencoders, graph neural nets, and of course the architecture that got me particularly excited in the field, reinforcement learning as well. Now, the interesting thing about reinforcement learning is it's not only applied to the protein folding problem and RNA folding problems, but it's actually also been applied for de novo drug discovery. One paper that really caught my interest was by in silico medicine that took drug discovery and de novo drug design and framed it as a Markov decision process problem. Now, the whole basis of reinforcement learning is you have an agent that's interacting in some environment, following a certain policy, and trying to optimize on getting the highest reward possible. And this is like in a game, you want the highest score. We can similarly create an agent taking actions to design the best drug candidate based off this idea of trying to get the highest reward in terms of optimizing for certain chemical properties, efficacy scores, as well as safety standards. And in silico's model in specific, the generative tensorial reinforcement learning agent, they've been able to bring 2 drugs to the clinical trial stage significantly cheaper and faster than traditional methods. Now, reinforcement learning is sick, but while I was researching this field, I got particularly interested in modeling drug discovery as not just any game, but a game of Sudoku, since it's my favorite. And with a quick Google search, I actually found out that people have tried this before. I came across a paper by UFT, University of Toronto, and what they did was they realized drug discovery and a game of Sudoku in particular are very similar. In Sudoku, you have these grids and you have rows and columns, and the whole goal is to take your numbers from 1 to 9 and place one of each such digit in each grid, each row, and each column, so you can't repeat it. And what this does is makes sure that each box each position near a specific grid is impacting each other. So you need to be mindful of the boxes around you. Similarly, in drug discovery, when you're building new chemicals, we have certain chemical rules we need to follow. Specifically in chemistry, the electrostatic forces, the whole idea of opposites attract and like forces will repel, right? And so what this specific research group did was train their model, their graph neural net, on Sudoku data and then fine-tuned it on their specific data, and it showed pretty well scores. But yeah, that's all pretty cool. There's a lot of different models being applied in the space, but this video in specific is going to dive deeper into how LLMs, large language models, which we've all seen with ChatGPT having insane results, especially in terms of generalization, and how we can apply that to drug discovery. So let's dive into it. So taking a look at those first initial steps of drug discovery, the design and make phase, there's 2 main ways in which AI can help researchers. Number 1, coming up with new drug candidates, that de novo drug discovery that we were talking about. But number 2, predicting chemical properties such that you don't have to run as many experiments and you're saving time and money. But the issue with current AI models being applied in the space is that there's too many options. There's so many different models that need to be used for different use cases, fine-tuned on different data sets, and it's confusing for the researchers. I mean, ever since ChatGPT has come out, we've seen how it makes our lives so much easier. I can have one model that I go to in order to fix my essays, but also teach me how to cook properly. So this whole idea of having one model, one universal model for biochemistry as well, can prove to be very successful in drug discovery, and hence why I've been interested in using large language models. These large language models not only enable us to generalize to different tasks like creating new molecules versus predicting properties, but it also enables us to look at different chemical properties and maybe start finding new drug candidates from scratch, but maybe even guiding it in a specific direction and constraining it to different chemical search spaces. So it just leaves the arena wide open. Now, not all large language models will enable this kind of generalization though. Large language models are based off this whole architecture known as transformers, which is based off of certain characteristics like having self-attention mechanisms and positional encoding. And if you're interested in, well, you should watch my previous video about transformers in order to understand their basic architecture first, because this video is just going to build on that transformer architecture. But what most researchers have been doing when applying LLMs to drug discovery is taking a transformer and then adding a regression head. And by doing this, they allow it to be very good at predicting continuous values, those chemical property values typically that we want. But then that completely destroys this generalization, because now we can't predict and create sentences for that generative molecular molecules that we want to do for the de novo drug discovery, and just coming up with better optimized molecules and drugs. And so I found one model in particular, the regression transformer from IBM, that actually takes this regression problem and frames it as a continuous sequence generation task instead. And by doing so, by using cross-entropy loss instead of typical regression loss functions like mean squared error, and it's able to train one model that's now able to both predict and generate new molecules, but also help in that property prediction task. So today I'm going to be analyzing the regression transformer. Let's get into it. This regression transformer works by first constructing an input sequence, which is constructed in a pretty particular way. It starts off with the chemical entities represented as smiles strings, property names as their own tokens, and property values as floating point numbers, with everything having separator tokens in between. Now that we have this input sequence, we can put it through the transformer, choosing to mask anything in between, and it will begin auto-filling or auto-completing that input sequence. And the great thing about this is now we can choose what we mask. We can map the actual chemical properties, or we can mask the chemical entity that smiles string, or we can mask half of the smile string and constrain it to that chemical search space. So it creates a very generalizable model that can do different tasks.
Over here, we can see the regression transformer's generalization capabilities. This multitask language model is able to handle both the numerical and textual inputs.
When deciding which transformer model architecture to use as the backbone for the regression transformer, the researchers had a couple different options. They could, one, use autoregressive models, kind of those GPT-like models, but there's a couple issues with that. Autoregressive models are trained in a particular way by passing one token into the model at a time. So you can only make predictions based off what you've previously seen. And if you think about it, if we're trying to predict the chemical properties, which are at the start of our input sequence, then we're not going to know any of the information that's shown later on in the input sequence, that molecule smile string, which is at the end. Instead, we could use something like a BERT-based model, which has bidirectional encodings, and so it basically learns a full contextual representation of the input sequence before it begins predicting the masked tokens. That's great, but another issue with BERT is they independently predict each of the tokens. So they don't have any context of one another. And that's why the XLNet backbone is used instead, which was proposed by Google researchers in 2019 as a substitute, a better option to BERT in order to overcome that limitation. And so the regression transformer model leverages this XLNet backbone as the core component of its architecture. Now that we have these engineered sequences, the next steps are tokenization and creating embeddings. Tokenization is the process of breaking down this input sequence into smaller chunks of tokens and then mapping these to learned embeddings, which are just vector representations of each token, keeping their meanings intact. So similar tokens will be having similar numerical values. They'll be closer to one another in this vector space. Now, this model in specific does something very interesting with the way that they learn their embeddings. The issue with taking regression tasks and modeling it as a sequential sequence generation task instead is that we're losing a lot of meaning that is in numerical values. This big data approach of just giving a model a bunch of data and telling it to replicate it loses hidden meaning in numbers. And so in order to bring this numerical meaning back into the model, they represent numbers in a very particular way using numerical embeddings to give that meaning back. Basically what they do is they take a number like 129, for example, and they'll give you meaning of what each digit is and which location in the number, what position it's at. So 129 will be mapped as 1_2 for that one value because 1 is the value and it's in the second place value. So the place values are ordered 0, 1, 2. And then you have the 2, which will be 2_1. And then finally the 9 will just be 9_0. And that way the model can actually learn what numbers are kind of being put in when the model gets the embeddings. And again, we need to give these models these embeddings and not those engineered sequences because underneath the hood, these AI models are just mathematical functions and they can only understand numbers. And then as for the actual alphabetic tokenization, they happen just as they would in any other transformer model. There are learned embeddings that are learned on a purely data-driven approach. The final model has about 27 million trainable parameters. And if you're interested about the actual architecture, check out my article or the actual regression transformer paper. But in general, it has its transformer encoders followed by the feed-forward neural networks and then the attention heads, as well as 20% dropout to make sure that the model's not overfitting on the data. And the researchers basically went ahead and created a synthetic data set by taking the Campbell data set and computing drug likeliness scores using the RD toolkit, and basically created more than 1.4 million molecules that they fed to our model and trained it on it. And what's really interesting are the specific objective functions that they defined. So they had one objective function for the regression tasks of predicting the chemical properties. They had one for the molecular generation tasks of creating SMILE strings that adhere to those primed chemical properties, as well as the ones that have drug-like properties. And then they had one self-consistency objective function that basically worked by taking a input sequence by masking the SMILE string at the end and priming it on certain chemical properties, predicting a molecule, and then feeding that back to the model, but this time masking the chemical properties instead, such that we can predict the chemical properties and then compute a loss on the original primed chemical properties and then the final predicted ones to make sure that the model is successfully adhering to those properties that we want it to adhere to. To assess the validity of the regression transformer model, the authors compared it to a couple different state-of-the-art models, and it actually showed some good results. So for the first task of predicting chemical properties, that regression task, it was compared to the over-benchmarked molecule net data set and specifically looked at 3 properties. And these 3 properties basically broke down to a chemical's ability to be absorbed within our body as well as their bioavailability. And the results were pretty good. The best baseline model, the XGBoost model, was outperformed by the regression transformer. And again, this is really interesting because you need to remember that the baseline model was optimized on predicting those 3 specific chemical properties, whereas the regression transformer could just as easily change the tokens of which chemical properties we're trying to predict, and it would do just as great. The regression transformer did slightly fall behind BERT-based and BART-based models that just had a regression head put on top, and it makes sense because they're both large language models, and they're kind of cheating because they're framing it as a regression task, whereas ours was using a cross-entropy loss optimization function. And because of that, we can also compare our model to benchmarks on that conditional molecular generation task. And so for that comparison, they were specifically looking at optimizing and creating higher log p-values based off a particular seed for a molecule. So this works by taking our SMILES token at the end of our engineered sequence and choosing to mask only some of the parts of the molecule and then putting in a higher chemical property value for the log p value. And then the outputs over here for this comparison will also very well. The regression transformer model beat a lot of state-of-the-art models in this domain. And again, you need to remember that the models it was being compared to were specifically trained to optimize for higher log p values, but our model could just as easily change this to optimizing for lower log p values, and it would work just as well. Okay, unfortunately, that's it about the regression transformer from me for now, but definitely check out my Medium article to learn more about the paper and the specific architectural choices. And also definitely be sure to check out the implementation of the regression transformer and the generative toolkit for scientific discovery. They've made it very easy for you to begin playing around with the model and maybe using it to accelerate your drug discovery workflows if you are a researcher. I've ran and annotated the Git repo myself as well, so check that out if you'd like some explanations on how to run the Python code and break that down a little bit easier. And yeah, that's it from me. I hope to see you in the next one.